So regulation of the tryptophan and lack operon involves negative control of genes because operons are switched off by the active form of the repressor. Next, now we'll study about the lactose metabolism in E. coli. Lactose is a secondary carbon source. Generally, glucose is preferred. Enzymes for lactose metabolism are lactose transport into the cell and then lactose hydrolysis into the monosaccharides. You can see the image. So, the lactose generally present outside the cell has to enter into the cytoplasm. So, in order to transport the lactose into the cytoplasm will happen with the help of glucose permease enzyme. And once the lactose enters inside the cytoplasm, the enzyme that is beta galactosidase, it is going to break down or hydrolyze into galactose and glucose. So glucose and galactose are the products of beta galactosidase. Sometimes the lactose converts into allolactose also with the help of beta galactosidase. Next, positive gene regulation. So sometimes in E. coli lack operon concept, you can see the positive gene regulation also. So where the control through a stimulatory activatory protein, that is CAP, that is catabolite activator protein. When glucose is cleared, the lac operon is activated by the binding of CAP. When glucose levels increases, CAP detaches from the lac operon and turn it off the system. So this is the regulation of lac operon. You can see how the operon is. That's the DNA. It contains lac I and then lac P and then lac O. These are all the regulatory gene. Lac I is a regulatory gene. Next to that is a PLAC, that is a promoter of the regulatory gene, so that the regulatory proteins will be synthesized. And after that you can see the operator gene, and next to that you can find the structural genes. So the three structural genes, they lac Z code for beta galactosidase, lac Y codes for permease, and then lac A codes for trans-acetylase. So beta galactosidase, we know very well that it will break down the lactose into glucose plus galactose and also allolactose. And the permease is going to help the lactose to enter inside the cell, enter the lactose into the cell. And trans-acetylase is an unknown function which needs to be understand in future. So next, how does a cell know which gene to express and which gene should stay silent? So when a gene is expressed, it is transcribed into mRNA. When a gene is silent, it is not transcribed. So what is an operon? Group of genes that operate together. Example, E. coli. So which ferments lactose? I already explained you about all these basic things. So in the lac operon, it regulates lactose metabolism. It turns on only when lactose is present and glucose is absent. So lactose is a disaccharide, a combination of galactose and glucose. So now you can see this. So generally what happens is in the operon, lac I is a regulatory gene which is going to synthesize regulatory proteins. So and this regulatory proteins may be a activator or repressor. So generally in E. coli lac operon system, the repressor proteins blocks transcription of lac genes. So what it does is repressor, it comes and bind to the operator. So once the operator is binded with the repressor and RNA polymerase cannot bind to the operator because already repressor protein is binded there. So because of that, the gene expression is stopped. That indicates the transcription does not happen. So because of that, translation is not going to happen and protein synthesis is going to switch off. You can see very well the RNA polymerase over here, it is unable to bind to the operator region. So now, since it is an inducible expression system, now inducer has to activate the gene expression. So what happens is the protein is especially the inducer, it will go and bind to the repressor. So due to that, structural conformation of the repressor protein changes and it releases out, and once that happens, the RNA polymerase is going to bind to the operator. So here you can see the gene expression, it got activated because the repressor moves away and the RNA polymerase, it starts transcribing, and once transcription happens, the translation happens and the protein synthesis will be happened. So the lac genes are turned off by repressor and turned on by the presence of lactose, which acts as an inducer. So when you see the lac gene expression, operon has two regulatory regions, that is a promoter and the operator. Promoter is a place where RNA polymerase binds and operator is a place where lac repressor will bind. So lac repressor when bound to O region prevents binding of RNA polymerase to promoter, turns the operon off. Lac repressor also binds to lactose. It has a higher affinity for lactose. When lactose present, lac repressor is released from O region, allows transcription of all the three genes. So this is the main concept of this lac operon, how the switch on and off mechanisms will happen in order to transcribe the protein. Okay, so in the next class I'm going to explain you about the tryptophan operon system. Okay.